Hello and welcome to Science View, where we cover the latest advances in Japanese science and technology. I'm your navigator, Tomoko Kimura, and this week's Science Watcher is Dr. Eiji Mizushima from the University of Tsukuba.
Hello, I'm glad to be here.
Here is today's lineup. On today's Leading Edge, we'll look at the front lines of treatment for thinning hair. A radical new treatment for thinning hair using cell technology is now opening up. This hair regeneration therapy may in turn enable regeneration of a variety of organs. And on J Innovators, Michelle?
Today I'll introduce a system that enables shop staff to perform instant calculations. The Takumi developed a system to instantly determine the total price of fresh baked bread using image identification technology. We'll learn about his technique and how he devised the system.
First, let's begin with today's Science News Watch, where we introduce one of the latest topics in the world of science and technology. Dr. Mizushima.
On May 9, 2016, with the participation of international research groups, including those from Japan, a research institution in Switzerland began large-scale experiments to recreate the conditions just after the formation of the universe in order to search for new particles that are unknown to humankind.
The experiments are being conducted at CERN, the European Organization for Nuclear Research, which is located in the outskirts of Geneva, Switzerland. The experiments involve use of CERN's particle accelerator, a giant experimental device with a circumference of 27 kilometers. In the experiments, researchers first accelerate protons that comprise matter close to the speed of light and collide them to recreate the conditions after the formation of the universe. Then search among the various particles that are subsequently produced for new particles unknown to humankind. The amount of matter discovered by humans up until now is thought to be only about 5% of the overall universe, so the remaining 95% is still a mystery. After improving the performance of their equipment in 2015, CERN was able to obtain data that demonstrated the possible existence of new particles that are still unknown. This led researchers at CERN to begin full-scale experiments, and they plan to repeat approximately 2.5 quadrillion collision experiments until October of 2016, with the aim of discovering new particles.
Approximately 6,000 researchers from more than 40 countries and regions all over the world are participating in the experiments, including about 100 Japanese members from research institutions such as Tokyo University, and together they are focusing on being able to understand the mysteries surrounding the origins of space.
And now let's move on to the Leading Edge. Today we will talk about cutting-edge research on thinning hair treatment. Dr. Mizushima, first let's start by looking at the mechanism by which hair is grown.
Okay, check out this close-up of the pores in the skin. In medicine, pores are called hair follicles. Dermal papilla cells lie at the root of the hair follicles and take on a role like that of a control tower. They stimulate the hair matrix cells that surround the dermal papilla cells and encourage cell division. In this way, the hair matrix cells that become old are pushed out. Let's see what that looks like.
You can really see them being pushed out. So why do people end up with thinning hair?
That has to do with the hair cycle, which is the cycle of hair being replaced. There are 3 periods in the hair cycle. The first is the growth period. After that comes the regression period, in which cell division declines. And then there is the rest period, in which cell division stops altogether. In most cases of thinning hair, the hair cycle is disrupted by movement of an enzyme in male hormone, which causes the growth period to become shorter. That's why research is underway on treatment methods to see if that growth period could be extended.
This is the research center of a major cosmetics manufacturer. They are developing a new treatment method using cell technology. Project leader Dr. Jiro Kishimoto is a veteran researcher who has been studying hair for over 20 years. Kishimoto and his colleagues are paying particular attention to the hair bulb hair root sheath cells below the dermal papilla cells. These are thought to be young cells as they surround the dermal papilla cells and eventually become dermal papilla cells.
Hair bulb hair root sheath cells stimulate the dermal papilla cells, and growth factor secreted from the dermal papilla cells causes the hair follicle to extend.
These cells are the key to extending hair growth as they activate the dermal papilla cells. First, hair follicles are harvested from the back of the head, a place where it is generally easy for hair to remain. Hair bulb and hair root sheath cells are extracted from the follicle and cultivated so that they can be increased. They are then returned to the skin. Consequently, these cells activate the dermal papilla cells and prolong the growth period, meaning that hair should increase. So what are the procedures in the actual treatment? First, they use a section of the scalp taken from the head. From there, the hair follicle in the root attached to the hair is harvested. Cutting the hair follicle reveals hair that has just been formed, which is the black part seen coming out. However, the little clump to the right of the screen is important. An upside-down view of that can be seen here. The protrusion at the top is the cluster of dermal papilla cells. The cluster at the bottom, which resembles the umbrella shape of a mushroom, contains the hair bulb hair root sheath cells. From there, we'll cut out the part that resembles the umbrella of a mushroom. Researchers then cultivate that part with original technology and increase the number without losing any of the characteristics. Then the increased cells are injected into the scalp. These cells move around the dermal papilla cells of their own accord. This stimulates and rejuvenates the dermal papilla cells, allowing them to return to normal, healthy function. The growth period of the hair cycle in particular is prolonged, and the hair is resurrected.
I hear concerns about thinning hair coming from people of all ages, so I think this treatment in particular can offer hope to those sorts of people.
Kishimoto and his colleagues are setting up a base in Kobe to gather more results about regenerative therapy and are working with medical institutions to continue their clinical research.
It's great that you can use your own cells for treatment.
I agree. However, this method can only be used for people who still have hair follicles.
Of course, that's a good point.
Some people do not have hair follicles due to the effect of anti-cancer drugs or injury, burns, or even just hereditary factors. The method we just examined cannot be applied to their case.
And that's why researchers are pursuing even more innovative methods.
Those innovative methods involve producing the hair follicle itself. Dr. Takashi Tsuji is conducting research into that kind of regenerative therapy.
We're considering how we can reproduce the process of hair generation. And that really is the key point to our research.
Tsuji has been focusing on two types of cells. One is the dermal papilla cell. This is the control tower for the hair follicle. The second are cells that are found in this part that is called the bulge area. Called epithelial stem cells, they can change into many different types of cells. These two types of cells have to meet in order for the hair follicle or organ to be made. Looking at a cross-sectional view of the scalp where hair has not yet formed after approximately 2 months have passed since the fertilization, the bulge area stem cells are here. The epithelial cells are the group that becomes the skin. They are lined up systematically side by side. Meanwhile, the dermal papilla cells belong to a group called the mesenchymal cells. A characteristic of these cells is that they can move freely between the other cells. In this period, two types of cells are summoned and meet each other. The hair follicle is first formed in the body, while these cells are communicating with each other.
These two types of cells react to each other, stimulate each other, and produce an organ that is the hair follicle. That's why the most important thing is to have these two cells adhere to each other densely and exactly.
In order to replicate the encounter between these two cells, Tsuji is repeating the process through trial and error. The result that he has finally arrived at is he used this collagen gel that has a high viscosity. Its properties are close to the composition under the human scalp. He injected a cluster of epithelial cells and mesenchymal cells into that gel. Since the gel has a high viscosity, the two types of cells do not spread out away from each other. In this way, he was able to create the conditions to have the cells densely connect with each other. This is close to the conditions when the two types of cells meet naturally. After cultivating the cells in this condition for several days, he placed them into a hairless nude mouse, and hair started forming after about 3 weeks. This is a cross-sectional view of an actual hair follicle that was created. The dermal papilla cells, as well as the system that includes oil-producing sebaceous glands, were regenerated. Moreover, the cycle of hair falling out and growing again was repeated in the regenerated hair follicle. In other words, the hair cycle was also recreated. That period is about 3 weeks, which is nearly identical to that of normal mice. Tsuji is now proceeding with his experiments on human cells and hopes to be able to apply his technology to a practical treatment within 10 years.
So an encounter between two cells is the key to hair regeneration. Why is this encounter necessary?
There is a kind of back and forth going on between the two cells. First, a protein called Wnt is sent from the epithelial cells to the mesenchymal cells. This is a command to form hair papilla. After that, a protein known as BMP4 is sent from the mesenchymal cells to the epithelial cells. This is the command to become the hair matrix cell and create the hair. The hair follicle is formed through the back and forth of this signaling.
It seems like the cells are stimulating each other.
Yes, and a mechanism like this does not apply only to hair, but to much more.
Let's listen to an interview with Dr. Tsuji from the Riken Center for Developmental Biology, whom we just saw earlier.
One point of our efforts for hair regeneration is that the hair follicle is really just a type of organ. In other words, we might be able to achieve regeneration of other organs in the future, not just hair. In the future, I would like to be able to create an entire liver. But that is very difficult at the moment. That is why we first have to start from something small, which is our efforts to regenerate hair.
We know that organs are formed from the resulting interaction after two cells meet. It's the same not only for small things such as teeth, salivary glands, and fingernails, but lungs and the liver as well. So researchers think that if we could apply this technique, we should be able to regenerate entire organs.
That's a very exciting thought, that studying the regeneration of hair may even lead to the regeneration of internal organs.
Yes, that's right. However, there are still challenges in regeneration of hair follicles. There is a limit to the tissue that can be taken from the human scalp. This will not lead to medical treatment if researchers cannot create 1,000 or tens of thousands of hairs from just one follicle. Since cultivation technology for epithelial cells has not yet been established under the present conditions, it will be necessary to clear that hurdle.
As a matter of fact, regenerative research on hair follicles using iPS cells is also currently underway.
Dermatologist Dr. Manabu Oyama is engaged in efforts to regenerate the hair follicles of body hair using human iPS cells. Oyama is examining the epithelial cells and keratinocyte that form the structure of the skin. We now know that it is beneficial if retinoic acid and protein BMP4 are added to iPS cells in order to allow these cells to form. This is how keratinocyte actually looks like when it's formed. 1 month has passed and the mature cells have formed at this point. Since keratinocytes are epithelial cells, there is a possibility that the hair follicle will form when they meet the mesenchymal cells. So Oyama came up with an idea there.
These are iPS cells that are in the middle of differentiating.
He used cells that were differentiating before the keratinocyte became a complete cell.
We found that the younger cells had a stronger ability to create tissue. Those young cells are taken from an adult and created. This is quite difficult. If we were to compare the cell to a boiled egg, the adult cells are like eggs that have just been boiled. It is better to create a cell that is like a soft-boiled egg. This is because of the possibility to shape it later. However, this can only be done with iPS cells.
Oyama combined the immature epithelial cells and the mesenchymal cells and implanted them beneath the skin of a mouse. As a result, they reconstructed this kind of structure that even includes the hair follicle. The hair follicle is concealed inside the object protruding down that looks like a needle. This is a hidden hair follicle. The green-colored part is derived from a human iPS cell. Although there is just a small amount, hair has been formed in the skin.
What we just saw was hair follicles regenerated inside the skin.
Yes, using immature cells is a key point to this research.
Does the fact that they succeeded with mice mean that they can try it with humans soon?
Unfortunately, that will require a little more time. They still have not been able to create mesenchymal cells from iPS cells in this research. That is why Dr. Oyama's research is using mesenchymal cells from mice. They will have to clear this hurdle in order to apply the technology to humans.
I see. Now, can you tell me about some research outcomes that were announced recently?
Yes, in March 2016, Yokohama National University announced that they succeeded in developing a mass culture of hair follicles. They used the cells of mice embryos. Before they can apply the outcomes to actual treatment, they will need to succeed with cells of adult mice and then human cells. But still, it can be said that they are making steady progress in their research.
One of the difficulties about cell research is that they have to consider so many circumstances.
Indeed. Even if animal experiments go well, that doesn't mean experiments with human cells will go well. Furthermore, this experiment using mice was for research on body hair. With humans, the type of hair on the head is different from body hair. So more research is needed to overcome that key difference and see whether or not regeneration is possible.
What other challenges do they need to overcome?
This kind of research is very new and is very much in the minority here in Japan. There are only less than 20 researchers in the field here. They would like more people to take an interest in this, so there can be a wider range of research. And of course, they want to be able to make it available for use as soon as possible.
We visited a mountainous area in Hyogo Prefecture's Nishiwaki City in the Kansai region. Hello, I'm Michelle. Today we're going to take a look at a checkout system that is being used in bakeries. Today we're going to meet a Takumi innovator who has developed an easy-to-use checkout system for freshly baked bread that cannot be barcoded or packaged. Let's go and see how the system works. We went straight to see the checkout system. As soon as the tray with the bread is set down, amazing! In just about a second, the breads have been recognized and all the prices are displayed. Gosh, that was so quick. Maybe even I can do it too.
Michelle, I think you can.
Yes, this is the Takumi who developed the checkout system, Kazuyoshi Tajika in person. The amazing thing about this system is that it's easy to operate. It can even be operated by new personnel. What do you have to do?
All you need to do is press this button.
Is that all? It's so simple. Above the tray is a camera which imports the image. The computer cross-checks the image with previously uploaded images of the bread, and using that, it deciphers the type of the bread and shows the price. When the bread is recognized correctly, the contour of the bread is marked in green. On the other hand, when the machine is uncertain, the contour is marked in yellow. In this case, you choose from several possible options on the monitor. If it thinks that there is only one piece of bread when there are actually a number of pieces together, you can cut them off from each other. When bread has a complex shape and the machine thinks that there are separate breads, you can make it into one piece using the magnet tool. The Takumi started its development with a request from a baker. Bread sounds far better when it is not packaged, but usually there are almost 100 different types of bread. The person who is in charge of the checkout system must know all the products that are sold, otherwise the checkout will not work smoothly. So Tajika and his team decided to make a system that is easy for anyone to use. He thought of using image distinction technology. But it was impossible to use the technology as is. Bread may come in different sizes or have different colors from the baking process. Although some bread may look similar, it could be a different type. The computer was not able to correctly identify that ambiguity. So then Tajika set his eyes on the information about size and shape that is used to analyze the image data. He then analyzed the image by turning it into a mesh. You can identify the size and the shape by the number of the mesh. And depending on the place of the mesh, it could differ in color and surface texture. By gathering more than 100 types of these kinds of information, the computer was able to distinguish a melon bun from among more than 20 characteristics. By using this method, the Takumi succeeded in distinguishing the difference of hard-to-tell breads. These are the prototypes that were made during development. 10 machines in 3 years. After numerous experiments on the height of the camera, the angle and lighting, the Takumi searched for the optimal state. This is the machine after many improvements, but there was a new problem.
The issue was that ambient light differs depending on the shop.
Ambient light is light in the surrounding environment that changes depending on time zone, weather, and lighting. The identification rate declined tremendously under the influence of different ambient light. So then Takumi thought of color correction. To do this white and black check sheet is used. For example, in the evening, when red light is stronger, color correction can exclude that red color. This is how the Takumi overcame the decline in the identification rate. Furthermore, this machine has a learning function, so the more it is used, the better its identification accuracy will become. Presently, the identification rate is 98%. The Takumi is now thinking of a way to use this identification system in hospitals to check the leftover meals of patients.
I think that it could be useful at a hospital because by knowing how much food the patients have eaten, hospital staff can figure out how much nutrition was taken in and use that information to improve treatment.
The operation is really simple. It seems like anybody could do it.
Yes, it's very simple. You only have to touch the screen once, so the burden on the shop staff is greatly reduced. And I heard that the staff now has more time to help the customers.
This image identification technology seems like it could be used in other areas besides bread.
Certainly, if research progresses, it could also be used in health management and safety systems as well.
Thank you very much, Michelle. So, Dr. Mizushima, how would you wrap up today's program?
Using the latest advances in regenerative therapy, incredible treatment methods to generate new hair may become closer to reality in the future. And there might come a day when we can regenerate more complex organs like the liver and the lungs. So I really think that regenerative research on hair follicles is a giant step in that direction. There are still many challenges and this research will take time. But I'm very interested in seeing how this research will unfold in the future.
Thank you, Dr. Mizushima. And that's all for Science View. See you next time.